Myself Professor Shamsul here. Welcome you all in today's session on plant physiology and metabolism, where we will learn about the gibberellic acid, another crucial regulator influencing plant growth. So, let us start. In the field of plant physiology, the historical background of gibberellins was initiated by Swada in 1920. Initially, he proposed the idea that infecting fungi, specifically Gibberella fusicori, might be secreting a certain substance. It was later in 1926 when Kurosawa successfully induced the Bakane effect in rice and maize seedling by treating them with the self-free culture medium derived from the growth of Gibberella fusicori. A significant turning point occurred in 1935 when Yabuta coined the term gibberellin for the active factor found in the culture filtrate of Gibberella fusicori. Building on this, Yabuta and Sumuki in 1938 not only isolated but also crystallized two biologically active substances and naming them as gibberellins A and B. Despite these milestones, the research in this field did not attract widespread attention until around 1950. Then a pivotal moment came in 1954 when P.W. Brain and his collaborator made a preparations from fungus culture isolating a distinct compound from Gibberella fusicori culture filtrate. This compound was named gibberellic acid, and in short it is GA, and gained universal acceptance as gibberellic acids. Another scientist, Radley, in 1956 reported substance resembling GA, and they were extracted it from the seeds and fruits of plants belonging to 9 different genera as well as from the shoots of pea seedlings. Now, as you know, the widespread occurrence of gibberellic acid in numerous species is established with their ubiquitous presence confirmed in both angiosperms and gymnosperms as well as certain other plant varieties. About 135 gibberellic acids have been characterized and 12 gibberellic acid have been isolated from Gibberella fusicori. Now we will discuss about the structure of gibberellic acid. Nearly all gibberellins are chemically associated with a broad class of naturally occurring compounds referred to as terpenoids. Terpenoids are constructed from 5 carbon isoprene units. Gibberellins are diterpenes composed of 4 isoprene units. As illustrated in figure, the fundamental structure of all gibberellins is based on the Ent-Gibberellinae framework. Furthermore, gibberellins from both fungal and plant were identified. They were systematically assigned numerical designation as gibberellin GAX, with X represented a sequential number based on their order of discovery. This is a figure of end-corene ring structure from which all gibberellins or you can say the gibberellic acids are derived. Certain gibberellins possess the complete set of 20 carbon atoms and are referred to as C20 gibberellic acids. In contrast, some gibberellins have only 19 carbon atoms, that is C19Gas, due to the loss of one carbon through metabolic processes. This metabolic alterations results in variations within the gibberellin molecule, influencing their biological activity and functions in plant growth and development. The easy and readily extraction of GA1 from fungal culture establishes it as the most abundant and readily available form of gibberellin for commercial use. It may be GA1, GA and GA4 as well as GA3. This accessibility carries practical significance, enabling its widespread application in diverse fields such as agriculture, horticulture, and the regulation of plant growth and development. Moreover, among gibberellins, these three, that is GA1, GA3 and GA4, are particularly noteworthy for their potent activity in promoting vegetative growth. These specific gibberellins play a crucial role in plant growth and development. Now, the structural requirement for GA activity. There are certain structural features influence the biological activity of gibberellins. One common characteristic is the presence of a carboxyl group at carbon 7 in all gibberellic acids, which is essential for their biological functions. Additionally, C19Gas with 19 carbon atoms are generally more biologically active than C20Gas, which have a complete set of 20 carbon atoms. Further, gibberellic acids with 3 beta hydroxylation, 3 beta 13 dihydroxylation or 1,2 unsaturation tend to exhibit higher activity. Notably, gibberellic acids with both 3 beta oxygen and 1,2 unsaturation displays the highest level of biological activity among gibberellins. These structural insights contribute to a better understanding of how a specific feature impact the effectiveness of gibberellins in regulating various physiological processes in plants. This slide shows the sites of GA synthesis. Gibberellins are most abundant in immature seeds and developing fruits. Interestingly, the gibberellin concentration tend to decrease to 0 in mature seeds. Arabidopsis, which you all know is a model plant species. There, the expression of GA20-OX is expressed in the apical bud and young leaves. This pattern suggests that these regions serve as the primary site for the synthesis of gibberellin in Arabidopsis. This slide possesses the information related to biosynthesis of gibberellic acid. As I have earlier mentioned, Gibberellins belongs to the class of tetracyclic diterpenoid acids. So, gibberellic acids undergo a complex biosynthetic processes in three different organelles. The synthesis of gibberellins initiates within plastid through the terpenoid pathway. Subsequently, these precursor molecules are subjected to modification in the endoplasmic reticulum and cytosol to yield biologically active forms of gibberellins. Moving to first step of gibberellic acid synthesis that takes place in plastids originating from glyceraldehyde 3-phosphate and pyruvate. However, in the endosperm of pumpkin seed, isopentenyl diphosphate is generated in the cytosol through the conversion of mevalonic acid derived from acetyl-CoA. Successive addition of IPP, that is your isopentenyl diphosphate isoprene units, leads to the formation of intermediates of 10 carbon compounds called geranyl diphosphate, that is the intermediates of 15 carbons called forensyl diphosphate or intermediate of 20 carbons called geranyl diphosphate, or in short you can say as GGPP. So, GGPP serve as the precursor for various terpenoid compounds including carotenoids and essential oil. Once GGPP is formed, the pathway becomes specifically oriented towards gibberellin biosynthesis. The pivotal step in this process is the cyclization reactions converting GGPP to N-choline. These reactions mark the initial gibberellin specific phase. The enzymes catalyzing these cyclization reactions are localized in the proplastids of the shoot meristem with no presence in mature chloroplast. Consequently, mature green leaves lose their capacity to synthesize gibberellin from IPP. Certain compounds like AMO1618, psychosyl and phosphon D act as specific inhibitors targeting this first stage of gibberellin biosynthesis. These inhibitors find application in agriculture to control plant height by restricting gibberellin production. The second stage of gibberellin biosynthesis involves oxidation reactions occurring on the endoplasmic reticulum, resulting in the synthesis of Ga12 and Ga53. Firstly, choline is transported from the plastid to the endoplasmic reticulum, where it goes oxidation to chorinoic acid catalyzed by choline oxidase associated with the plastid envelope. This is succeeded by a contraction from 6 to 5 carbon rings, ultimately yielding Ga12 aldehyde. Further oxidation transforms Ga12 aldehyde into Ga12, making the production of first gibberellin in this pathway. Additionally, many plant gibberellins undergo hydroxylation at carbon 13 position, giving rise to Ga53 from Ga12. All the enzymes involved in these transformations are monooxygenases that utilize cytochrome P450 in their reactions. Importantly, these P450 utilizing monooxygenases are localized on the endoplasmic reticulum. So, now we talk about certain compounds such as paclobutrazol acting as inhibitor of P450 monooxygenases, specifically hamper this stage of gibberellin biosynthesis before the formation of Ga12 aldehyde, thereby serving as growth retardants in plants. Coming to the last stages in the gibberellin biosynthetic pathway, which occurs in the cytosol and involve a group of soluble dioxygenases. These enzymes rely on 2-oxoglutarate and molecular oxygen as co-substrate, utilizing iron 2 plus and ascorbate as cofactor. In most plants, two primary chemicals modification takes place. First one is hydroxylation at carbon 13 on the endoplasmic reticulum, or carbon 3, or both, and the second modification is successive oxidation at carbon 20. The final step of this oxidation process involves the release of carbon 20 as CO2. So, when these reactions involve gibberellins initially hydroxylated at C13, the resultant gibberellin is Ga20. Ga20 is subsequently transformed into the biologically active form, that is Ga1, through the hydroxylation of carbon 3. This figure is a schematic representation of all three stages of gibberellin biosynthesis I have discussed earlier. Now, we are moving about the transport of gibberellins. They are produced in the shoot and have the capacity to be transported systematically throughout the plant via the phloem. Additionally, intermediates of gibberellin biosynthesis may undergo translocation through the phloem. Notably, the initial stages of gibberellin biosynthesis can take place in one tissue with subsequent conversion to active gibberellins occurring in another. Moreover, the presence of gibberellins has been detected in root exudates and root extract, suggesting that root possess the capability to synthesize gibberellins. Furthermore, these synthesized gibberellins can be transported to the shoot through the xylem, emphasizing a potential bidirectional transport system between roots and shoots. This slide discusses about the mechanism of action of gibberellic acid. As you know that during the germination of grains, hydrolytic enzymes are generated to facilitate the breakdown of stored carbohydrate, fats and protein reserve. The induced synthesis of these enzymes by gibberellic acid has offered valuable insights into the specific mechanism through which gibberellic acid exerts its effect. A total of 29 hydrolytic enzymes are present, of which amylase play a very important role. So, I have to stop here and the rest will be continued in the next lecture about the physiological role of gibberellic acid as well as that of the signal transduction pathway. So, in continuation to my previous lecture where we have discussed about the gibberellic acid structure, biosynthesis and mechanism of action,